This is going to be the first of 3 podcasts on the digestive glands. We're going to have a podcast on the liver, one on the gallbladder, and one on the pancreas. So this first one is going to deal with the liver. Now just a little bit of a review. The liver, gallbladder, pancreas, along with the major salivary glands, are usually considered extrinsic digestive glands. They're considered extrinsic digestive glands because they're outside the wall of the GI tract. These organs produce enzymes, buffers, emulsifiers, and various lubricants, and the products of these glands and organs are delivered to the GI tract by ducts. These organs also produce hormones and a variety of blood proteins. There are several clinical correlations that are useful to keep in mind. We're not stressing them here, but it is useful to be aware of them. So first, there are some correlations with the salivary glands, usually the parotid gland, and I'm not going to talk about them here. I just list them for you to read them. With respect to the liver, cirrhosis is an extremely important problem clinically. Various hepatotoxic substances, alcohol, barbiturates can interfere with liver function. We can have jaundice, either an obstructive or a hemolytic jaundice. In your clinical training, you're going to learn a lot about liver chemistry and how that relates to the various organelles. I'll show you a slide or two about liver enzymes. Clearly, viral hepatitis, Hep A, Hep B, Hep C are important clinically. Gallbladder, the most usual problems are gallstones, cholecystitis, and most of the gallstones turn out to be cholesterol stones. And then the pancreas, things like pancreatitis can be a life-threatening illness. Carcinoma of the pancreas is fairly significant. Less than 50% of the patients diagnosed with pancreatic carcinoma survive more than a year. And obviously type 1 and type 2 diabetes are almost reaching epidemic proportions in this country. So this is just a brief overview of some clinical correlations for all of the digestive glands. Your learning objectives that would relate to this podcast and the next 2 for the liver, gallbladder, and pancreas, we want you to learn the histology of these organs. For the liver, you should be able to describe blood and bile flow as related to histological organization. We want you to become familiar with 3 alternative histological views on liver organization. This is going to be important as you go forward in pathology. We would like you to understand the cell biology of the hepatocyte as it relates to liver function, and you'll recall we had an earlier podcast on the hepatocytes. You might find it helpful to review that podcast. For the gallbladder, we'd like you to be able to describe how bile is formed, as well as being able to identify the gallbladder histologically. And for the pancreas, as well as being able to describe it histologically, you should be able to describe endocrine and exocrine functions of the pancreas, relate how the cell biology of the acinar cell relates to exocrine function, and how the cell biology of the islet cells relate to endocrine function. And again, there was a podcast on the acinar cells of the pancreas. You might find it helpful to review that as well. With respect to the liver, I'd like you to understand that most of the blood from the liver is supplied by the portal vein. Portal vein, of course, is a vein that's between the capillary beds of the GI system and the liver sinusoids. And then 20% of the liver's blood is supplied by the hepatic artery. And so just a question that you should think about, which blood going into the liver has the higher oxygen tension? This diagram just shows how material goes from the GI tract to the liver. So it's just schematically showing the GI tract and the liver putting this circuit in for the blood circulatory system. And you know that most material absorbed through the GI tract. If you go down the tract, this just kind of shows where in the GI tract major things are absorbed. Most of these materials get into the portal vein and will get to the liver through the blood vascular supply. But of course, fat-soluble vitamins, and cholesterol gets to the liver through the lymphatic channels. I remember the lacteals that we talked about in the intestinal tract. Pancreatic enzymes that are going to empty into the duodenum, and bile that's going to empty into the small intestine. And remember, bile has a dual role. It's involved in the digestion of lipids because it's an emulsifier, and it's also a vehicle for excretion. Here is a brief overview of liver function. I'm not going to test you on this for our course necessarily, but it is something that you should try to keep in mind as you integrate what you've covered in biochemistry, what you're going to cover in physiology, and as you go forward in your clinical practice. All of this is very important. As you understand, the liver produces most of the circulating plasma proteins. So, for example, molecules like albumins, various lipoproteins like VLDL, LDLs, HDLs. The liver produces glycoproteins, for example, haptoglobins, transferrin. The liver produces lots of clotting factors, for example, prothrombin, fibrinogen. I think you already appreciate that the liver is involved in storage and metabolism of vitamins and iron. The liver degrades toxins and xenobiotics, essentially makes foreign substances water soluble. You understand that there's oxidation of xenobiotic material in the smoothie iron mitochondria by the cytochrome P450 system. A lot of these products get conjugated with glycuronic acid, glycine, or taurine, and you should remember some of this from biochemistry. You clearly will remember from biochemistry the central role the liver plays in various metabolic pathways. So, for example, glucose, glycogen, and cholesterol metabolism. Clearly, the liver is involved in beta oxidation of fatty acids and ethanol conversion to acetaldehyde. That particularly is important with organelles like peroxisomes. The liver is involved in the production of ketone bodies, which are used by the heart and brain. And the liver is probably playing a role in modifying hormones, and clearly the liver is involved in bile production. So this is an important overview of some of the liver biochemistry. When you look at the liver anatomically and histologically, the liver is covered by a connective tissue capsule called Gleason's capsule. The liver lobule is the functional unit of the liver parenchyma, and in this diagram the liver lobule takes on a polyhedral shape. And if you go to the higher power, you should note the arrangement of the hepatocytes kind of in rows or in plates of cells. There's a falciform ligament, which is part of the ventral mesentery, that connects the liver with the abdominal wall. If we talk about the portal vein, we can equate the portal vein to an interlobular vein. We can equate the hepatic artery to an interlobular artery. We can equate the bile duct to interlobular bile ducts. We can also mention that the sublobular vein is going to drain blood from the central veins in the liver, and these sublobular veins are shown like this. This profile of the liver lobule, the functional unit of the liver, points out some important anatomical landmarks. So, for example, we can define a portal triad. The portal triad has a branch of the hepatic artery, a branch of the portal vein, and a branch of the bile duct. We can talk about sinusoids running like so, and we can talk about the central vein. In this classic liver lobule, the central vein is at the center of the lobule, and these portal triads, I would like to call them portal areas, are at the periphery of these lobules. And this is just a histological view of the portal area of the liver. So you can see a branch of the bile duct, a branch of the hepatic artery, a branch of the portal vein like so distended, and then right here is a branch of a lymphatic vessel. So portal triad we think of classically as hepatic artery, portal vein, bile duct, but there's also a lymphatic vessel. So there's four structures in the triad, which is kind of an unusual nomenclature. So I like to just say that this is the portal area. Note that the hepatocytes are arranged in rows or plates of cells. Here again is the classic liver lobule, blood flowing from the periphery, from the portal area, towards the central vein. The portal vein and the hepatic artery empty into a common space. That's called the sinusoidal space. The liver sinusoids are lined by an endothelium, and atrial and venous blood mix in the liver sinusoids. Bile flows in the opposite direction of blood flow in the liver. So bile flows from the hepatocytes towards the portal area. This is just a higher power diagram showing hepatocytes. Things colored in orange color would be the bile canaliculi. These are spaces between the hepatocytes into which they secrete bile. These bile canaliculi are coalesced into branches of the bile duct, which would eventually make their way into the portal area. These bile ducts are called Herring's canal, and it's interesting, the ductal epithelium in these bile ducts may be a source of stem cells for the liver. Now, I also would like you to take note on this diagram. The artist has purposely drawn it this way to show some hepatocytes as binucleate and some hepatocytes that have nuclei that are different sizes. That actually is important because some hepatocytes are actually binucleate and some hepatocytes are actually polyploid. Now, I'd like to go over 3 alternative descriptions of the liver, and these are important histologically, they're important physiologically, and they're important from the point of view of understanding liver pathology. The first description that I'm going to talk about is the classic liver lobule. We've already mentioned that. The classic liver lobule would be what's outlined like this, these hexagonal type structures. This emphasizes endocrine function of the liver. The central vein in the classic liver lobule serves as the morphological axis of the lobule, and at each point on this structure there are portal areas. So the classic liver lobule emphasizes endocrine function of the liver. The portal lobule, which is shown as this triangle, emphasizes exocrine function of the liver. It has as the morphological axis the bile duct, which is in the portal area, and at each point on the triangle is a central vein. So this is emphasizing exocrine function. And then finally, there's a liver acinus represented by this diamond-shaped structure. It emphasizes blood perfusion, and it describes different zones of metabolic activity. So in this liver acinus, it's also called an acinus of Rappaport, there are several zones that can be described. There's cells that are in zone 1, there are cells that are in zone 2, and there are cells that are in zone 3. At 2 points on this diamond are the central vein, and at 2 other points are the portal areas, and we come to appreciate that cells in zone 1 have the highest level of oxygenated blood because those cells are closest to the branches of the hepatic artery. Cells in zone 2 have less exposure to oxygenated blood, and finally cells in zone 3 have the least exposure to oxygenated blood. You might also think which cells in which zone are exposed to more toxins, and you'd have to think about this from the point of view of how is material being supplied to the liver. Here's in one slide the importance of understanding the 3 concepts of liver organization. So the classic liver lobule, the portal lobule, the liver acinus. The classic lobule, as we said, emphasizes endocrine function of the liver. The morphological axis is the central vein, blood flow from the hepatic artery and portal vein to the central vein. The portal lobule emphasizes exocrine function of the liver, morphological axis, the portal area, actually the bile duct of the portal area, bile flow from the hepatocytes to the central bile duct. And the liver acinus, or acinus of Rappaport, emphasizes liver pathology. The morphological axis is actually the boundary of 2 classical liver lobules. So cells in zone 1 are the first to receive oxygen and nutrients and toxins. They're the first to show changes after bile duct occlusion. Cells in zone 1 are the first to regenerate. Cells in zone 3 are the last to receive oxygen. Therefore, they're the most sensitive to ischemia. They're the least sensitive to most toxins. So the liver acinus, importantly, emphasizes liver pathophysiology. As you go forward in pathology, it's going to be really important that you remember the liver acinus in describing liver histology. This slide shows a liver biopsy from a patient with congestive heart failure. There's ischemic necrosis, which is very evident in the cells that are in zone 3. These cells show multiple round vacuoles that indicate lipid accumulation in the ito cells, and there's phagocytosis of dying hepatocytes. Hepatocytes that are in zone 1 are fairly normal, and those that are in zone 2 are only showing minor morphological changes. This cartoon shows 3 different surfaces on hepatocytes. There's a hepatocyte to hepatocyte cell surface, so that would be a surface like this, hepatocyte to hepatocyte cell surface. The hepatocytes would be joined one to another by desmosomes. There's a bile canoniculus that can be described. This bile canoniculus is bounded by tight junctions, and this would be the exocrine surface of the hepatocytes, and the bile canoniculus is actually equivalent to the GI luminal space. You have to think about that anatomically, but you should appreciate that it's equivalent to the GI luminal space. And then there's this perisinusoidal space of Disse, which is the endocrine surface of the hepatocytes. This is a surface that's bathed in plasma, and the cartoon shows labeled a sinusoidal lining cell. That is essentially an endothelial cell of the liver sinusoids, and the cartoon wants to show a red blood cell in the sinusoidal lumen. There are also Kupffer cells that line the sinusoidal lumen. We'll talk about those in just a few minutes. This is another way to look at the hepatocyte. So we've shown the hepatocyte drawn diagrammatically this way. Here are the endothelial cells of the sinusoidal capillaries. Remember, these are capillaries that don't have diaphragms. There's a discontinuous basal lamina. Everything colored in yellow is plasma space. So I want you to understand, here's plasma space. Here's the space of Disse between the endothelial cells and the hepatocytes. And notice that the space of Disse actually runs in continuity between the hepatocytes, but it stops at these tight junctions that bound the bile canoniculi. So this green space here or here is the exocrine surface of the liver. All of this yellow space would be the endocrine surface of the liver. Now, it should go without saying that within the lumen of the sinusoidal capillaries are all of the formed blood cell elements. So the red blood cells, all of the white blood cells, platelets, etc., those formed blood cell elements cannot get between the endothelial cells, so there are no formed blood elements in the space of Disse, but otherwise the space of Disse contains all of the plasma proteins that would be in the blood, that would be in the lumen of the sinusoidal capillary. So the diagram makes it very clear that the hepatocyte is completely bathed in plasma except for the spaces where there are tight junctions separating the bile canoniculi from the remainder of the perisinusoidal space of Disse. A little bit of the cell biology of the liver. Remember, the endothelial cells, we've just talked about them, line the sinusoidal capillaries. The sinusoidal capillaries are fenestrated. They're discontinuous. There are no diaphragms. There's no basal lamina. There's a little bit of collagen type 3 because the basal lamina is discontinuous. Kupffer cells are actually monocyte-derived cells. These are resident macrophages in the liver, and they form part of the sinusoidal lining. They are in close juxtaposition to the endothelial cells in the sinusoidal capillaries. Ito cells are also called hepatic stellate cells. These cells store fat and store vitamin A. They're thought to be a pericyte-like cell. They may actually be a stem cell in the liver. These cells synthesize retinol binding protein. They play a significant role in fibrinogenesis. They synthesize collagens type 1 and type 3. They contain contractile elements, so some people think they contribute to portal hypertension, and it's thought they may organize extracellular matrix material during liver regeneration. And then hepatocytes, of course, are the functional or parenchymal cell of the liver. These cells are involved in most of the metabolic activities of the liver. Some hepatocytes are binucleate, some are actually polyploid, so they can be 4C. Some hepatocytes actually go up to 8C, 16C, 32C, even rarely up to 64C. Hepatocytes have extensive rough ER to synthesize proteins for export, and for membrane proteins they have extensive smooth ER for detoxification of xenobiotics and lipid biosynthesis. Hepatocytes have abundant mitochondria and peroxisomes. You'll also notice a lot of lipid droplets and glycogen deposits within hepatocytes. Here's another diagrammatic view showing some hepatocyte, some bile canoniculi like this, a large ito cell, and the endothelial cells lining the sinusoidal capillaries, and here an example of a Kupffer cell. So here's the lumen of the sinusoid. This would be the space of Dissay here. Here's an electron micrograph of a hepatocyte. So here would be the sinusoidal capillary, the lumen thereof. Notice the space of Dissay. Notice there are some microvilli, not elaborate microvilli, but the surface of the hepatocytes do project some of these microvilli to increase membrane surface area. So here, the space of Dissay, as shown here, would have all the plasma proteins within it, but none of the formed blood cell elements. Notice the numerous mitochondria that you can see in the liver. You can see rough ER. Some of these electron-dense materials would be glycogen deposits. Here's a higher power view of a hepatocyte. Notice the large lipid droplets. Here is the nucleus. Here's the nucleolus. I'm going to very carefully trace some cells here. Here are the boundaries. There's one hepatocyte and another hepatocyte here, another hepatocyte like so, and then on the bottom of the field a hepatocyte. So allow me, if you will, to trace very slowly the cell boundaries on this large hepatocyte. So I've traced one there. Here I'm tracing another cell boundary like so, and I'm tracing another cell boundary like so. What I want to point out, let me trace this boundary between this cell and this cell. I'm tracing this boundary, and then look, this space is a bile canoniculus like that. Here again the cell boundary. So there is a bile canoniculus. Now this is very hard to appreciate from this electron micrograph, but the space shown right here where this asterisk is marked, that's actually perisinusoidal space of Dissay. So let me go to this cartoon again. Here I was in this bile canoniculus, junction between two cells, perisinusoidal space of Dissay. So you can see that here in the cartoon. Here's the perisinusoidal space of Dissay. Here's the bile canoniculi. Here's a higher power view, a really very high power electron micrographic view. Look at the bile canoniculus, look at the tight junctions here, and then the desmosomes holding these hepatocytes together. Here, like this, and here, like this, would be perisinusoidal space of Dissay. Look at it's being a little more elaborate here. Look at the large profiles of rough ER. Look at the mitochondria. Some of these electron densities are probably lysosomes. There's lots of glycogen deposits that you can see in these cells as well. Here's a very high power light micrograph. It's one cell thick plates of hepatocytes that are surrounded by sinusoidal capillaries. Bile canoniculi surround the hepatocytes, and they're shown in parallel sectional plane at the arrows. So here I can trace bile canoniculi around these hepatocytes like that, and then the arrowheads show the bile canoniculi in cross-sectional planes. Here is a view here and here of endothelial cells. These endothelial cells are lining the space of Dissay. Here is a Kupffer cell, and here is an ito cell. The image on the left shows the cellular machinery for protein synthesis and carbohydrate storage. So you can get a sense just looking at the cartoon that glucose brought into the hepatocytes, converted to glycogen, stored in the hepatocyte. When there's a need for glucose, glycogen is broken down and it's released. Amino acids brought in undergo protein synthesis, going through Raffier to Golgi, packaged into secretory vesicles. The material will be exocytosed, and usually most exocytosis in the liver occurs by constitutive secretion, so the vesicles will be coated with COP2 proteins. The diagram on the right is just exploring the exocrine function of the liver, so bile secretion and bilirubin metabolism, just highlighting, for example, bile acids that are reabsorbed in the intestines, get brought into the hepatocytes. Some of these bile acids get excreted into the bile canaliculi, so they'll get recycled back to the GI tract. Bilirubin forms from the breakdown of hemoglobin as red blood cells age, enters the hepatocyte. Glucuronate transferase conjugates water-insoluble bilirubin into water-soluble forms so that it can be easily excreted by the hepatocytes. This slide is for reference only in relation to some of the lipoproteins involved in the liver. Lipoproteins are, of course, complexes of protein and lipids, are important in transporting cholesterol and triglycerides in the blood. As you know, cholesterol and triglycerides do not circulate freely in the blood. They cannot remain in suspension. They have to complex with lipoproteins, and that will decrease their hydrophobicity. You should recall from biochemistry there are 4 classes of lipoproteins. Chylomicrons are the lightest of the lipoproteins. Remember, they're synthesized in the small intestine and they transport absorbed fat from the small intestine to the liver. Very low density lipoproteins are high in triglycerides. These are synthesized in the liver and to a lesser extent in the small intestine. These transport triglycerides from the liver to other organs. Low density lipoproteins are elevated. Circulating levels correlate with increased risk of cardiovascular disease. These are assembled in the plasma and to a lesser extent in the liver. These transport cholesterol from the liver to other organs. And then the high density lipoproteins correlate with decreased risk. Elevated levels of the high density lipoproteins correlate with decreased risks of coronary vascular disease, and of course the ratio of HDL to LDL is important. HDLs are assembled in the plasma and to a lesser extent in the liver. HDLs transport cholesterol from the peripheral tissue back to the liver. And I put this slide here just for your reference. These are things you should have learned in biochemistry. You'll become more familiar with these as you advance in your training. Remember, this slide talks about LDL receptor gene mutations, which can account for high circulating levels of LDL. We went over this in an earlier podcast, so I'm not going to go over it here again, but you can remember that there can be various classes of mutations in the genes that lead to the assembly of LDL receptors. Mutations in these genes lead to familial hypercholesterolemia. This is the most common Mendelian disorder. Heterozygous individuals will show familial hypercholesterolemia. Homozygous individuals usually don't survive beyond the second decade of life because these individuals really can't metabolize cholesterol to any great extent. The LDL genes are on the short arm of chromosome 19. Liver enzymes and liver function, again, something for your reference. I'm not going to expect you to know too much about it for our course, but it is important for you to appreciate some of these enzymes. Alanine aminotransferase is a damage marker. It usually indicates hepatocyte cell death, and it will increase with hepatocyte cell death. Normal values are shown in parentheses. ALT will be low in liver failure. Aspartate aminotransferase, or AST, is a damage marker. Usually it's increased with alcohol-induced hepatocyte cell death. It will be low in liver failure as well. Alkaline phosphatase is an obstructive marker. If ALT and AST are at normal levels and ALP is elevated, that might indicate that there's an obstructive disorder in the liver. Gamma glutamyl transferase confirms liver disease because ALP can be elevated in other disorders. GGT may vary a little bit with age and with sex. You've talked about bilirubin. Elevated indirect or unconjugated bilirubin might indicate a deficiency in hepatocyte function, as the liver can't conjugate bilirubin. Elevated direct or conjugated bilirubin oftentimes indicates an obstructive disorder. Serum albumin measures normal liver function. If albumin is very low, that might suggest there are liver problems. And clotting time or prothrombin time, if it's elevated, that might suggest that there's liver malfunction or liver damage. Just a histological overview of the liver. So here you can see the portal area. I think for sure there's a branch of the hepatic artery, a branch of the bile duct, probably a branch of the portal vein, and that might be a branch of a lymphatic vessel. That might also be a branch of a lymphatic vessel in the portal area. And here is the central vein. A high power view of the central vein. You can see the rows or plates of hepatocytes, and you can see the liver sinusoids. A nice branch of the portal area, so clearly branch of the hepatic artery, branch of the bile duct, branch of portal vein, and perhaps branch of a lymphatic vessel. I've circled this area because I want to highlight for you this hepatocyte that has two nuclei in the cell, and I also want to highlight, look at the size of these nuclei and compare them to this nuclei. This nucleus is clearly polyploid in relation to these nuclei, because this one is clearly at least twice as big as these nuclei. And here I highlight an ito cell. I won't expect you to pick out on a histological image the ito cells. I won't expect you to necessarily recognize on a histological image that some of the cells are polyploid or that they're binucleate cells, but I would like you to know that fact. It's a higher power view of some of the plates of hepatocytes. So this is sinusoidal space. Here are endothelial cells. Look, here's some white blood cells in the lumens of the sinusoids. Space of Dissay would be shown like this. And these white spaces that are in these hepatocytes, some of this would be equivalent to the perisinusoidal space of Dissay. These arrows are actually showing junctions of bile canoniculi. If I were to trace like this, you can see boundaries between hepatocytes. Here's boundaries between hepatocytes right here. You can see a bile canoniculus highlighted here at the black arrows, a bile canoniculus highlighted here, bile canoniculi. No problem that you can clearly trace some of the boundaries on these hepatocytes.